Hello, good morning, friends. In the previous classes, we are discussed about the Marselia, especially the sporophyte structure and the morphology of the Marselia and the anatomical structure of the root, petiole, rhizome, and the sporophytes, the leaf that we have studied, and that continue with the reproduction. So in case of the Marsilia, they are reproduction by the vegetatively as well as asexual mode. So usually the asexual reproduction or the vegetative reproduction is takes place by the formation of tubers, especially in case of Marsilia when uncertainty of the water condition or the scarcity of the water condition, what happens? So usually in the rhizomes region, that will itself to swell and to form a food storage material. Those are known as the tubers. So when these tubers are get separated from the mother plant, they will be capable to produce a new plants. So that's the one type of vegetative reproduction in case of the Marsilia, especially in case of Marsilia hirsuta and Marsilia minuta involved in the tuber formation. So going to the important aspects of Marsilia reproduction, that's a reproduction by the spores. So in case of the Marsilia, the is a heterosporous form. Already the Marsilia is a pteridophyte and it is a heterosporous. That means they produce the two kinds of spores. One is a microspore and another one is a megaspore. So usually these megaspores are produced in a separate sporangia and they bond to the special bean shaped bodies called as a sporocarp. The sporocarp is nothing but the spore producing structure in case of the Marsilia. So usually Marsilia have the special characteristic, that's microspore, and the megaspores will be produced in a special structure called as a sporocarp. So usually going to the structure of the sporocarp, so this figure is showing the how actually the sporocarp will be there. Usually the sporocarp is ovoid nut like structure and attached to the basal part of the petiole with the help of stalk. So usually in the early stages it is a green softened structure, so and when matures it will turn to the dark brown in color. So usually the sporocarp number may be varies, so usually 2 to 20 sporocarp produced in a plant. So usually the sporocarp attached to the petiole, so that may be also varies and have the peculiar structures. So just look at these diagrams how actually the sporocarp attached to the petiole in different species of the Marsilia. In the first diagram you will find the attachment of sporocarp in the Marsilia polycarpa. That means the more number of sporocarp will be produced, and in second one is a Marsilia quadrifolia, third one Marsilia minuta, and fourth one the Marsilia vestitica. So like that, the in each species the attachment of the sporocarp may be varies. So in the first it is diagram, it's showing that the more number of sporocarp attached to the different position of the petiole. So in the last you will find only the nodal region that produces single sporocarp. So like that the sporocarp number may be varies and the attachment may be the varies. So this is actually the structure of the sporocarp. The sporocarp already I told, there's a bean like structures will be there and it is a simple structure and having the stalk, raphe, tubercule and the center body. The sporocarp which differentiate into mainly the stalk of, that is a pedicel, and a body. The stalk is fused laterally to the back of the body of the sporocarp. Generally these refer as a raphe. So sometime may be asking question that what is raphe. Raphe is nothing but the body that is attached to the sporocarp and they have the distinct ridges in their position. So usually upper distal portion end of the raphe is consist of the two teeth like structures will be there. Those are called as tubercules. Sometimes they may ask question, where do you find tubercule? In the sporocarp of the Marselia you will find the tubercular structures. So going to the anatomy or the cross section of the sporocarp, that's varies. They consist of sporocarp wall, vascular system of the sporocarp, and usually the section, that's the horizontal, vertical, and the longitude, that's HLS, VTS, and the VLS, that may be varies in the structure. So this is how actually the sporocarp wall will be there. The wall of sporocarp is very hard and thick, and sometimes may be a highly resistant to the drought condition. So usually they consist of different regions, that's the epidermis and the hypodermis. So usually epidermis is made up of the cuboidal cells, and the hypoderm consists of the two layers of the radially elongated palisade parenchyma layers, and inner parenchymatous layer is gelatinous. They will help to sporocarp and proper protection, and in the center portion that is the supply of the vascular tissues. So usually the single standard enter into the stalk, extended to the raphe, and then passes along the dorsal side of the sporocarp. This standard is called as a dorsiventral bundles, and this give out the number of the traces on both faces of the sporocarp, and these traces extended downwards and to form a specialized structure called as a lateral bundles. One is a dorsal bundle and another one is the lateral bundle. In the figure you are looking that, that's the lateral and the dorsal bundle. The number of position is varies with the different species based on the sori, and it may be sometime dichotomous branching, and usually the portion of the dichotomous small branches arises, that is called as a placental bundle. And this is the HLS view of the sporocarp. So that is the sporocarp with the horizontal longitudinal section that consists of the upper portion with the stalk bundles, and lower portion you will find the gelatinous ring will be there, and usually in the center you will find the indusium and also the sorus. That's very important. So sori are seen in the two alternative rows. Each sori has a receptacle which contains the one terminal megasporangium and two microsporangium on the lateral side, surrounded by a specialized structure called as a indusium. So that's very important. What is indusium? That's a specialized structure to protect it to the microspore in the Marselia HLS view. The sori overlap each other, and indusia of the adjacent sori are partially fused. So this is actually the VTS, that's a vertical transverse section. That's the first one, HLS and VLS and the VTS. So usually the HLS and the VTS is that's a vertical transverse section that may be little bit varies, and the lateral bud extended from the dorsal side of the to the ventral side of the sporocarp can be seen in this type of the section. And this is one is a VLS, vertical longitudinal section of the sporocarp, and here you can find the gelatinous ring, and this is how you can peculiarly spot the sori in a different angle. The either microsporangia or megasporangia very easily visible, and this is how the enlarged view of these pictures. Okay, so usually when we look at the structure and how actually these spores burst out from the sporocarp. So usually the dehiscence takes place with a different mechanism, but usually when they in case of the Marselia, so when it's open or the split, just like in dicotyledons what the cotyledons split out and they will be sought out, so like that same thing will be occur in case of the Marselia. So usually the sporocarp is split out, and through that the spores will be dehiscent, that's the microspores. This is the structure of the male gametophyte. That's Marselia is already I told, that's a heterosporous and producing a two types of spores, that's the micro and the megaspores. So usually the microspores are referred to the male gametophyte and megaspores is refers to the female gametophyte. So usually the microspores consist of the androcytes, that is a metamorphosis, just like a, so it look like a screw. So when a natural screw will be there, because the head region and the ring like region will be there. And usually similarly the megaspores, that's the larger size spores, that's the female gametophyte. This is the structure of the megaspores, and usually they have the neck canal and the ventral canal cells, and the center region you will find the megaspores, that's a female gametophytes, that's the center region consists of the. Usually the fertilization after when the microspore and megaspores get the mature, so undergoes to the fertilization in the female gametophyte. That's one of the sperm fuses with the egg cells to form the diploid zygote. The zygote mother cells of the next sporophytic generation develop into a young sporophyte within 2 to 4 days of the fertilization. So this is how the life cycle of the Marselia. So usually the first, that's the vegetative reproduction by the tuber formation or the fragmentation of the rhizome. They will be generally vegetative reproduction takes place. So in case of the sexual reproduction, first they produce the sporocarp, 2n in number. The sporocarp consists of the microsporangium and the megasporangium, and the microsporangium produce the microspores, that is in haploid in number. Megaspores also haploid in number, that is called as a male gametophyte. Megaspore is called as a female gametophyte, and the male gametophyte produce the antheridium and they produce the antherozoites. The female gametophyte produce the archegonium, they produce the egg. When these antherozoid and the egg, they will fuse to form a fertilization. Once the fertilization occurs, so usually the oospores will be formed. That oospores is a 2n, that's a diploid in number. So when these oospores split into germinate and produce the germ tube, and new Marselia sporophyte will be produces. The first half portion, that is from meiotic portion to the fertilization, usually the haploid in nature, and the later all position will be diploid in nature. So this is how the actually the complete life cycle of the Marselia. So first, so when this is a sporocarp, this is a plant body, they produce the sporocarp at the basal region, and these sporocarp produce the micro and the megaspores and undergo to the meiotic division. The first portion is called sporophyte. The second, when the spore producing region is called as a gametophyte phase, and usually the gametophyte phase during the meiotic portion, they undergo to the micro and the megaspores will be produced, that's the male and the female gamete. They undergo to the androcytes, and the egg cells will be formed to form a oospores. These oospores are the diploid in nature. So whatever you are find, that's a sporophytic region is almost diploid in nature, and the gametophytic region is a haploid in nature. So this is the complete life cycle of the Marselia. So in the today's class we are discuss the reproduction in case of that's the vegetative or asexual mode, and the sexually mode by the formation of spores, and usually the different structures and the sporocarp, especially the sporo HLS view and the transverse section, longitudinal section view of the sporocarp, and these have the different structures and the life cycle of the Marselia that we have studied in the today's video lecture. So usually for the exam point of view, I already told there may be some 1 marks question, what is indusium and what is the sporocarp, and what is the HLS view, what is the vertical section view. So like that the usually general question. For the 5 marks question, generally explain the HLS of sporocarp of Marselia, that will ask, or sometime maybe a chance of getting the explaining the megaspore structure and the microspore structure for the 3 marks. So this is an overview of the Marselia structures. So in the previous classes we are discussed the complete general account of the Marselia from the morphology to the anatomy. So today's class we are discuss the reproduction in case of the Marselia. So I hope you all got the concept. So if did not concept, ask the question. Thank you for your patience listening.